Hello everyone, this is the stop where we're going to talk about some bean research. I'm going to talk for a little bit about some of the things that I have going on. Then I'm going to pass the microphone over to Dr. Bao and his team to talk about things that he has been doing out here in this field. And then you're also going to hear from my graduate student Ekat Hampton about her work. Oh dear. She says she's fine. She's trying to get you handouts because we made these nice handouts, but then they didn't hand them out to you. Okay, so hopefully everybody survives. Okay. So first I want to talk to you about our snap bean variety trials. We've been doing snap bean variety trials for a number of years, and that's because we have increasing numbers of growers producing snap beans for processing and for fresh market. The biggest problem with growing snap beans in this region is heat stress. When we have high night temperatures, we don't get very good pod set, and or we get pods that are misshapen and deformed. So the main focus of the snap bean trials has been to identify varieties that have heat tolerance. So we had one of the varieties that has performed well in past years that were heat stressed is a variety called PV857. That is from a company that used to be called Popfreen, but now it has another, it's like KWS or something. That company has a new variety, which was the top yielding variety in our trial that we planted at the beginning of June 6th, and matured during heat stress. So these are some plants from that variety. We harvested these over 2 weeks ago, so that's why there's some dry pods on here. They're not green beans anymore, but this is a variety called Raptor. Other varieties that performed well under heat stress are Greenback, which is a variety that is meant for fresh market, and PV857, which I already mentioned, and then there are a couple of numbered lines that haven't been released as varieties yet, but like I said, the top varieties were Raptor, Greenback, and PV857. We have another trial that's actually the last thing planted in this field that is our non-heat stress trial. It was planted mid-July. Another thing that we have going on with beans is I am part of a project where we're looking at producing Shelly beans. Has anybody heard of a Shelly bean before? Okay, so a Shelly bean is kind of a regional term for common bean that is the same species as green beans and black beans, kidney beans, cranberry beans, navy beans. They're all the same species, but these are harvested at the succulent stage, the same stage where you would harvest a green lima bean, which we're much more familiar with in Delaware. So these are some plants that I pulled out of the border row. This is a cranberry bean variety. This is a type that is very typically used for Shelly beans, not necessarily in the US, but in other parts of the world. But we are looking at 18 different varieties. It's a regional project, so there are other sites in Virginia where they're looking at the same 18 varieties, and we have varieties from the kidney, cranberry, Great Northern, navy, black, and pinto classes in that trial, and they're all, and so that's down there where the leaves are starting to turn yellow. That's the Shelly bean trial. These are varieties that were bred for dry bean production, so we are trying to see how they would work for succulent stage production. I wanted to give you a little bit of an update on some things going on with the lima bean breeding program. I am part of an ongoing project with collaborators in California and South Carolina to understand the physiology and genetic factors that are related to traits that we're really interested in, like heat stress, nematode resistance, the upright plant architecture, seed maturity, and then something that's been really interesting as a part of that project is the quality evaluations. We sent 40-some samples of frozen lima beans out of the trials here to UC Davis, and they did culinary or sensory evaluations of those lines, and then this fall we'll be sending larger amounts of samples to do consumer preference tests. When you get a handout, there's some pictures and stuff showing the diversity of some materials that we're developing to use for genetic studies, and that is this block right here. And if you grow lima beans, you're probably tired of hearing about me talking about the breeding program and wondering when you can ever grow any of these things. So the update on that is that the green-seeded baby lima types are being increased by Pure Line Seeds. Maybe they'll have some available for trial next year. That's what they have told me. They're hoping. I have some non-green seeded types. So reds, speckles, things that the processors generally don't want around here that are being increased by Commonwealth seed growers in Virginia for more of the home garden or specialty market. Whole lima beans, they're way up there. I have been providing seed to growers in the Mid-Atlantic region to trial. And for Fort Hook lima beans, I have some good stuff, but seed production of that type remains difficult. If you want to hear more about our nematode work in lima beans, you should come to the field day on August 28th. If you need details, you can talk to me later. Okay, now I'm going to pass the microphone to Dr. Bal, and he is going to tell you about all the cool, technology stuff.
Hello, everyone. My name is Bao. I'm from Plant and Soil Science Department. My research focuses on digital agriculture, specifically high-throughput plant phenotyping. So we're using sensors, robots to help breeders like Dr. Ernest to measure plant characteristics. She needs to evaluate many different traits like plant height, architecture, or flowering time, and also yield. So we're trying to make that process easier for her. So we're using ground robot and drones. So on this ground robot here, we have 3 cameras, one at the top and 2 at the side. On the side, so they're capturing the RGB color image of the plants, and also it can capture the 3D model of the plants as well. So Ashish, is the video playing? Okay, so yeah, so if you look closer, you can, the monitor shut down, but you can see what the image look like or video look like, and the depth image and 3D point cloud. So from the videos, we're hoping to detect flowers and measure architecture and also count pods, number of pods. Hopefully that will have a good correlation with the yield and help use that data to select the high yield and early flowering varieties. And over here we have a drone. Right now on the drone it has a LiDAR sensor, so that can capture a 3D point cloud of the field, and that data can be used to measure the plant morphology, like plant height, the width. And we also have a hyperspectral camera that can measure many hundreds of colors in the visible and near infrared spectrum. So in a regular camera, you can only see red, green, and blue, but this will measure over 100 wavelengths in the visible and near infrared spectrum. So that gives you very detailed information about plant health and their physiological conditions. So that data can also be useful in terms of selecting the high performance varieties here.
Thank you, and we are looking forward to seeing if we can make things work faster in the breeding program. Okay, so I'm going to hand this off to Katerina Hampton, who is a graduate student in plant science.
Thank you. Hi, everyone. I'm Katerina Hampton. I also go by Ekat, if you know that one. So I'm working on lima beans in the greenhouse over there, actually. I have a diversity panel of 150 diverse lines from all over, mostly the United States and South America. It's comprised of lines from the University of Delaware as well as UC Davis. I'm taking a look at the effects of extreme temperatures, so particularly high nighttime temperatures. So that's when it affects the flowering stage the most, is when it's hottest at night. So I'm taking a look at stressing them out during the flowering stage at 80 degrees at night, and then the consistent temperatures during the day of whatever that may be in the greenhouse. So I'm taking a look at kind of the process from pollination all the way to germination, from germination again to pod set, etc. So I'm taking a look at like the whole process. So on your handouts, you can see some of my recent work. I'm currently looking at pollen. So I have done stigma collection, collecting the female part of the flower, and I've stained them red, and then I've taken images of them under the microscope and taken a look at pollen counts during our control, which we grew them in March. And with the cooler temperatures, and then now we have them in the greenhouse flowering at high nighttime temperatures, taking a look at them currently, what they're looking at for their pollen counts. And then one thing in particular we're looking at is stigma exertion. You can see that in the images below. So high nighttime temperatures can affect the shape of the flower, and that misshapen image of the flower, as well as stigma exertion. So the stigma will exert past the anthers, and it will not produce a pod because it's not being pollinated. So I'm taking a look at the physiological effects as well as the genetic effects. So I'm taking a look for specific markers in the genes that might identify specific parts that might affect like stigma exertion or seed filling. So I'm taking a look at the process both genetically as well as physiologically.